Hello, my juniors. Welcome to another biochemistry lecture series. It's your great science tutor Lorenzo, and today we'll be doing gene therapy. So we want to work on gene therapy today. You get it now. So in my last class we did molecular probe. So we have done molecular probe. The next one is what? Gene therapy. So after gene therapy, I'll be doing biotechnology. So I'll be moving like that. You get it now. So if you are new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video, you'll be sure to be notified. If you want me to make a video for you, what will you do? Drop it down in the comment section. I promise I'll make that video for you. You get it now. So let us jump right into the video. Are you getting now? So the introduction, what is a gene therapy? What is gene therapy? So gene therapy is a treatment technique. Hold it. I want you to underline it one by one. Gene therapy is a treatment technique used in the treatment and the prevention of diseases. So it is a gene, we call it gene therapy. Naturally, gene and therapy, which means using gene to treat somebody. So I want to use gene to treat somebody. It's a treatment technique used in the treatment and prevention of diseases by adding, removing, or altering the gene sequence in the patient's cell. Are you getting me now? So gene therapy is using gene to treat somebody. So you can either treat somebody by adding, removing, or altering the gene sequence. Are you getting me now? So hope you have gotten it like that. So when you use gene to treat somebody by what? Adding to his gene, are you getting that? Complementing his gene rather. So by deleting one of the gene, or by doing what? Altering anyhow. You can alter it anyhow. Just alter the gene just to help the patient. You get it now? So now if we look at the type of gene therapy, we have what we call somatic gene therapy, we have what we call germline gene therapy, and we have what we call gene augmentation therapy. Okay, now like I said, in this one, it's the future of science is in what? Molecular biology. In gene is the future of science. So if you are good at gene, that means you are good in science, because this is the future. Because we have cancers, we have so many diseases that are untreatable. So with all this molecular biology like gene probe, biotechnology, this is the future of medicine and science. So it's very, very important. You should know how to do it. So the first one, which is somatic gene line therapy. In somatic gene line therapy, this is a type of gene therapy in which the gene is introduced to a somatic body cell. The gene is introduced to a somatic body cell. So if we introduce the gene into the somatic cell, we are calling it somatic gene therapy. Okay, now so now why is somatic gene therapy good? One, it is not inherited by the offspring. So if I'm using it for a patient that has not given birth, what happens? Even though I alter the patient gene and he or she have not given birth, it cannot affect the child, because in somatic gene therapy, if I alter the somatic cells, it cannot be passed on. I guess number two, it is currently used in medicine. Why? Because it is used in medicine because it is not inheritable, and since it's not inheritable, because if it's inheritable, it's bad. You get it now? So the last one is used to treat various diseases and conditions. You get it now? So it is not inheritable. So introducing the gene to the somatic body cell is for somatic gene therapy. So how does it work? How does it work? One, you have to have a normal gene. So I've mapped out a patient gene. And there's a part of the gene that has an alteration. There's a part of the gene that has an alteration. So since I know that this patient gene in this particular line has an alteration in the somatic cell, I introduce a normal gene. Are you getting me now? I'm just trying to complement. So if I introduce a normal gene, what will happen now? That gene I've introduced will be used to form protein. And what happens? The patient condition will improve. It's so simple. Yeah, so we just use this to treat diseases and various conditions. Why? By taking a normal gene, a normal complementary gene is not, it can be from anybody, but make sure that the patient, the gene you want to introduce is complementary to the patient, just like a molecular probe. So if you take a normal gene, you put it into a patient's somatic cells that has been altered in some other way. So if it's altered, what happens? Once I input it there, what happens? It starts making protein by translation. So it will first replicate, transcribe, and now translate. So I'm just using it to treat the patient for that while. Nothing happened. So that part of the gene cannot be inherited. You get it now? But in germline gene therapy, what happens? I'm introducing the gene to the germ cells. You get it now? So if I introduce the gene to the germ cell, which is the sperm and the ovum, what happens? It can be inherited, because in fertilization, the sperm must meet the ova. If the sperm meets the ova, it makes a growing zygote. You get it now? So that can be inherited. But if I introduce it to the somatic line, what happens? It cannot be inherited because I'm just using it, introducing, there's a default in the patient what somatic cell. Maybe it could be due to that default, that's what's causing diseases. So if I introduce, what happens? The patient will be fine. You get it now? But in germline gene therapy, what happens? If I introduce it to the germ cells, what happens? It can be inherited. Are you getting me? Since it's inherited, what happens? You cannot just do it because anytime you have just altered the gene and you want the patient to be transmitting the gene, so it is not used in human. Again, it's not used in human, and it is prohibited because of ethical what consideration. So because you are using, instead of using, the goal of gene therapy is to treat somebody. So if you now alter the gene and you now alter the gene causing what genetic mutation and that can be transferable, it is not nice. So it is not used because of ethical what consideration. So what I do, I prepare the gene. I get him. I introduce it to the sperm and ovule. So if I introduce it to the sperm and ovule, it forms an ova. So that if I introduce it to the sperm of ovum, what happens? It will form an embryo. When it forms an embryo, it can be passed on to the offspring, and that is bad. It's just that you are trying to play God by forming something, and that is also the future because there are so many people that is undergoing that research currently. Another one is what gene augmentation therapy. Are you getting now? So what is gene augmentation? Because we say in somatic, we are introducing it to the somatic cell. In germline, we are introducing to the germ cell. In gene augmentation, what happens? This is a type of gene therapy in which a normal gene is introduced into the cell to compensate for a defective or missing gene. Finish. So you are using this one now in the word gene augmentation, which means the patient has a cell, a half or a partially good cell or a defective cell, and I now introduce. So when I introduce, what happens? I am doing what? Augmenting with the patient's own. So the patient's gene is defective, and I use a normal gene introduced in order to augment it. So it's just like somatic gene therapy, but in somatic gene therapy, the target is the somatic cell. But this one is the target is in that defective part of the gene, a defective part or a partially damaged gene. So when you augment, you use a normal gene to augment for it. You get it now? So one, you have to have a defective gene. Before you can do gene augmentation, you have to have a defective gene. It's either a defective gene or a missing gene. So when you have it, you now introduce a normal gene there. So if we introduce a normal gene, what will the gene do? It will undergo replication, transcription, translation, and what? Then it will now form a new protein. You get it now? So very, very simple. So I've explained the somatic gene therapy, I've explained the germline gene therapy and gene augmentation therapy. So if we are to know the quick difference between them, first of all, so somatic gene therapy is to the somatic body cell, germline gene therapy is to the germ cell, which is the sperm and the ovum, and gene augmentation is to what? Cell with defective gene. You get it now? So somatic cannot be inherited, only germline can be inherited, and also gene augmentation is what? It cannot be inherited. So I told you that somatic gene therapy is just like the gene augmentation. Are you getting me now? So also based on human use, you can only use somatic gene therapy and gene augmentation, but you cannot use germline. And the ethical issue is more in the germline than in augmentation and somatic. You get it now? So now the next one here is mechanism of gene therapy. What is the mechanism of gene therapy? Because they can ask you in your exam. So number one, I have to identify the defective gene first. Identify the defective gene you want to you want to use a new gene to treat. I identify it. It could be bad, it could be missing, it could be diseased. So you identify the defective gene. I get it. If you identify the defective gene, what happens? So you now prepare a normal and functional copy of that gene. Are you getting me now? You prepare it now because the first one is that you identify the defective gene. So you now check the complement system of that gene to now you now make a copy of it. You get it now? Because I want to use my own gene to do what? To what? Treat. So it's just like molecular probe. Now I'm making a gene complement in myself. Are you getting me? So I'm making it because I want to use that gene I make, just like a molecular probe. But a molecular probe is used to identify. You get it now? So it's just like the concept of a molecular probe. So I check the faulty gene. What do? I make a normal and a functional copy of that gene. Then do you know what I do? I insert. I now insert this gene using a vector. But before I do, I have to prepare a vector too. There have to be a vector. Vectors are what are molecules or are pathogens that helps me carry my product. So they help me carry and deliver the gene into the normal genome. You get it now? So I have to have a vector in hand. A vector will help me to deliver. So I first of all I identify the defective gene. Two, I prepare the normal and the functional gene. And the normal and functional gene to be a copy of that gene. Then when I am done, do you know what I do? And I put it inside the vector, and I allow the vector, inject the vector into the system so that the vector can help me locate where I want to replace and want to treat. So the vector will deliver the gene. And when the vector delivers the gene, what happens? There will be formation of a new gene product. Are you getting that? There will be formation of a new gene product. So what with the new gene do? Just like a probe, we go and identify. But in a molecular probe, you are just inserting it to look for where it is complement to. That is the molecular probe. But in this one, you already get a copy of it. You have already known what the copy is. Then you now do what? Insert it. Let the vector carry it to that part. So when the vector carries it to that part, what happens? It will represent the new gene. Are you getting now? You do what we call topoisomerase. It will ligate or remove the defective gene to topoisomerase. Then when you now do him, it will now insert itself end to end. It will insert itself end to end to that defective part of the gene. When it inserts itself end to end to that defective part of the gene, what happens now? You can use it to make what? Product. Product in the sense that the gene will now start working normal. The gene in that particular organ or the body cell that is damaged will now start working properly. And how will it start working properly? It will start what? Forming what? New products. You get it now? So first of all, identify the defective gene, prepare a normal functional gene. Now, when you prepare a normal functional gene that is the same copy, you now insert it into a vector. When you insert it into a vector, the vector will deliver the gene to the target site. First of all, by doing what? Binding end to end. I get it now, using a topoisomerase. Binding is end to end, which you call the DNA gyrase. Binding end to end, and the work is to what? Make new product for that system. And when you start making new products, the disease will be what? Corrected. You get it now. So look at the flowchart. Defective gene, I form a normal gene, I insert to a vector, I deliver the gene by the vector, and new product will be what? Formed. So now, what is the method of application here? How can you apply gene therapy now? Number one, are you getting me? You have a viral method and a what? A non-viral method. So we are just looking at the vector you want to use. Remember, I told you that a vector is used to deliver. So you can use a viral method and a non-viral method. A viral method mean that you are using a virus to introduce. The virus is the what? Is the vector. But the virus doesn't mean it's a what? An active virus. It's an attenuated virus, which means the virus cannot cause what we call inflammation. Even though it causes mid inflammation, but it will not cause major inflammation. You get that? So they are using normal viruses which you are immune to. So now, what are we using? What type of virus? We are using a retrovirus. We are using an adenovirus. And we can use other viruses. So if we are using a retrovirus, we are using it to integrate into the host genome. Are you getting now? It is used for long-term or gene expression. So we are using a retrovirus. A retrovirus is just like an antivirus you use to treat viruses like HIV and AIDS. Are you getting that? So when you are using a retrovirus product to what? Insert the new gene, what happens? You are using it to what? Integrate the whole genome and do what? A long-term gene expression. You can use an adenovirus, but an adenovirus doesn't integrate the host genome. Are you getting that? It doesn't integrate the whole genome. It can only infect dividing and non-dividing cell. So if you want to get a permanent gene expression, you use a retrovirus. But if you want to it to affect a dividing and non-dividing cell, you use what? An adenovirus. So retrovirus does what? Integrate the host and use for long-term gene expression. While adenovirus, it cannot integrate the host. It's just bind to what? Dividing and non-dividing cell. Other viral vector you can use is what? Adeno-associated virus, herpes simplex virus. You can use, but all these viruses are what? Attenuated. Hey, they cannot cause diseases. They are just in form of vaccine. They are attenuated. So if they enter the body, you only need high amount to start causing disease. And also, we have gotten immunized. You also use it in a patient that is immunized because at least they are immunized against those viruses. Now it is used depending on the target cell you want to pursue. Are you getting now? Is used. So I told you can use a retrovirus, an adenovirus, an adeno-associated virus. You can use a herpes simplex virus. So if you want to use a non-viral method, are you getting now? If you want to use a non-viral method to deliver the gene, example of a non-viral method, you can use a naked DNA. A naked DNA, a liposome, and you can use what you call an electroporation method. Okay, now, so a naked virus, now in your mind you say, what is a naked virus? When you are using a naked virus, you are just directly injecting what you call a plasmid DNA into the cell. A naked virus, you just take a plasmid part of the DNA and you inject it into the cell. So using a plasmid DNA and injecting it into the cell, that is a naked virus. You are not using the full product. You are just taking one part of the virus, the inside part of the your inside part of the DNA rather, and you are injecting into the cell. So that is a naked virus. It is simple and less expensive because it's hard to bring out the virus and prepare it. You just use the plasmid part of the virus. Now, the next one is liposome. For you to use a liposome, what happens? A liposome is a covering of a DNA. Are you getting me? Because DNA is covered by what? Lipid vesicle. Are you getting me? It's covered by lipid vesicle. So what happens? So if you are using a DNA liposome helps to protect DNA and aid delivery. So even though you are using any virus, you have to cover the viral product with a liposome. So a liposome helps you to deliver the DNA successfully. So first one, you can use a naked DNA virus. You can use a naked DNA, which is the plasmid part of the DNA. You can use a liposome. A liposome is the covering of a DNA using what? Lipid vesicle. This one you use is the electroporation method. In the electroporation method, electro means you are using what? Electricity. So you are using electrical impulses to create pores in the cell membrane so that the DNA can enter into the cell. When we say the DNA is the source, the form gene you want to use to treat. The form gene you want to use to treat. Remember, gene are encoded in what? DNA. So for you to deliver a DNA sequence into a person, it has to be, for you to deliver a gene into a person, it has to be contained in a DNA sequence. And that DNA sequence has to be covered with what? Liposome. Are you getting now? And also you can use electroporation method. The electroporation method, what happens? You are using what? Electricity. Electrical pulses, not big one. A low frequency pulses to create pulse inside the what? Cell. So when it creates pores inside the cell membrane, what happens? DNA can pass through the cell membrane to the target cell where it has to what? Bind. So you can use the viral method of vector, or you can use the non-viral method of vector. The viral method of vector is using what? The retrovirus, adenovirus, adeno-associated virus, herpes simplex virus. So, but this one has their own disadvantages. So, but you can use a or you can use a non-viral method, which is the naked DNA, a liposome, or the electroporation method. So now, what is the application? What is the uses of what? Gene therapy? One, you can use it for cancer. Gene therapy can be used to target and destroy cancer cells or stop their growth. So what happens now? Maybe a cancerous cell has been proliferating too much. Then you now take a good DNA, and a good gene and you send it into the body for the purpose of doing what? Inhibiting the continual growth and destroy the cancerous cell. You can use in what? Sickle cell anemia. Right. Remember, in sickle cell anemia, your body is producing sickled red blood cell. Sickled red blood cell doesn't pass through the blood vessel very good. Do you know what? It has to start attaching to the blood vessel because it cannot pass successfully. So that is sickle cell. So now what happens now, due to maybe there's manipulation of the of the DNA sequence. So if there's manipulation or there is a mutation, especially in the hemoglobin part of the DNA, what happens? It causes sickle cell anemia. So you can use it to do what? Help to treat by what? Inhibiting or going to bind to that particular part, that particular defective part, or that particular DNA that has been mutated. So when you use the gene will remove that side. When you remove that side, it will not bind and the patient will be fine. Are you getting me? So you can use it in hemophilia. So what is hemophilia? Hemophilia is a what? Is a blood disorder where patient bleeds uncontrollably. So when it's bleeding uncontrollably, why? Because it cannot make what? Blood clot or factors. Blood clot or factors. You get it now? So the patient cannot make blood clot or factors, so he bleeds excessively. Any injury like this will cause a lot of bleed. This is also due to defective and mutation, genetic mutation in the gene that codes clotting factors. So you can also use gene therapy to inhibit that mutation. Cystic fibrosis now. In cystic fibrosis, what happens? There's a increase in mucus secretion in the lungs. So the mucus secretion increases the surface tension of the lungs. What happens? Preventing air from going to the blood. So there'll be a high blood air barrier due to sickle cell anemia. So here there's defectiveness in the gene that codes for what increase our normal functioning of the respiratory system. Here, the mucus production will be high. Chlorine channel will also be what's mutated. So you get it now in sickle cell, in cystic fibrosis rather. So you can also use it to treat cystic fibrosis. Also, you can use it to treat neurological disorders like what Alzheimer's, dementia. You can use it to treat what Parkinson and others. You get it now. So what are the challenges in gene therapy? One, it is expensive. Anything genetic research is expensive because the machine, even the delivering tools, the one you use to follow up, all of them are just expensive. And also, there are always failures. Are you getting me now? So especially in treating sickle cell anemia, so many research has come and so many clinical trials have done, but no one is useful. Even cancer too has not been treated, but they just do chemotherapy and blast radiation, but there's no adequate because there are always failures. Because sometimes the gene we can fail. Now it induces immune response because when you are using viral method, retrovirus, adenovirus, and other viruses, it can cause immune response and the body will start attacking it, causing inflammation, and it can be bad. Unpredictable side effects. So you can start it now and start injecting DNAs or genes into the body. And when you start injecting all this, what happens? It can even destroy other genes, and causing what more harm than a good thing you want to do. So it can cause unpredicted side effects. Also, it can destroy other functional genes. I've just said it. If you can introduce the gene by vector and instead of it go and binds to another gene, binding to another gene, destroying that gene. You get it now? Also, it has short duration of action. So all these genes you are introducing doesn't last long. It's a short stuff. But using a retrovirus, is used to do long-term gene expression. So, but most of the gene therapy are short while. So you can do it inhibit for a short while, dropping the cancerous growth or dropping the infection, but later it will come back. Get it now? So sometimes you can do the one you do now and maybe inhibit or you are treating and patient will die because now it can cause blood vessel constriction or blood vessel dilation or inhibit blood flow to the brain or stop oxygen. It can cause anything. It can cause death. Ethical issues. Are you getting that? Ethical issues can also be there, just like germline therapy. Sometimes they don't want it because you can be playing God, and sometimes you can change the dynasty of a patient. Maybe a patient parents are good, or the patient is good. Are you getting me now? But when you did the gene therapy, it was fine, but you start causing a lineage worth of cancer. Maybe the individual just don't have cancer. It was just coming to get treated for a particular disorder. Then due to gene therapy, you now start mutation, and the patient has not married. So when the patient give birth, the cancer gene can start from them. So it poses an ethical concern. Also, all these things you use it in human cloning. So you can use it to clone, bring another person into this world. That and it defies the normal rules. You get it now. So it's also like skilled professionals. So they are it's not all professional, like I told you. Those people that are skilled in all this molecular biology and gene research, they are very few. You get it now. They are very few. So that is why most of this research have not been done. That's why we need more people that will go there. Are you getting me now? So I hope it all makes sense to you and you have understood everything I've taught in gene therapy. And what that gene therapy is, using gene to what? Profile treatment and inhibit disease by either adding, removing, or what? Augmenting. You get it now? So you have somatic gene line therapy when you add the gene to the somatic cell. You have germline gene therapy when you're adding it to the germ cells. And you have what they call gene augmentation therapy when you have a defective gene and you're adding it. So if you are new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video, you'll be sure to be notified. If you want me to make a video for you, drop it down in the comment section. I promise you I'll make that video. So I'll be seeing my next video in biotechnology. Keep watching me, keep sharing my video. Bye bye.